Okay, well, hello everyone, and welcome to the third and final presentation of our November webinar series. My name is Beth Melius. I'm the Foodborne and Enteric Disease Program Manager at the Washington State Department of Health, and also co-director of the Washington COE, and I will be your host for this session. The title for today's session is ATG GAC, What Can Sequencing Do for Me? Before we get started, we wanted to let you know that this webinar is being recorded and will be made available for viewing later on our website. We will also make these slides available to you. If you registered for the webinar series, you will be emailed when the recording is available. So we are so pleased to host you all today. The Washington State Integrated Food Safety Center of Excellence was established as a CDC COE, supporting the western region of the U.S. and Guam in 2019. Our COE is co-directed by myself and Janet Baseman from the University of Washington School of Public Health. So today our training will focus on why subtyping matters, sequencing technology, analysis methods, interpreting phylogenetic trees, and it is primarily for public health staff, both epi and environmental health, responding to foodborne outbreaks. Here are our learning objectives for this session. If you have any questions during the presentation today, please use the Q&A function in Zoom in the toolbar. We will be monitoring the Q&A and we'll take questions at the end of the session. And with that, I'm pleased to introduce our speaker for today. Cassandra Allen is a molecular epidemiologist at the Washington State Department of Health. She earned a Bachelor of Science in Microbiology from the University of Idaho and a Master of Public Health in Epidemiology from Virginia Commonwealth University. Cassandra has worked in applied epidemiology at both the local and state level and has worked with enteric disease data since 2013. She is excited about the transformation of enteric disease surveillance as lab methods have evolved from PFGE to sequencing and enjoys sharing her passion with other epidemiologists. So welcome, Cassandra, and take it away.
All right, thank you, Beth. Well, I'm excited to be here today, and here's our outline of what we'll be going over. So first I want to start with why subtyping matters and really how it can be used and why we care about these technologies. So I want to start with a story, and this is the story from my first job in public health, which was actually in Virginia. And I had only been on the job a few months. I was covering all general communicable disease for a multi-county region. And I got a call from the state health department from the central office, and they said that one of my salmonella cases was a PFGE match to a national cluster. And I wasn't quite sure what that meant at that point. But they asked me to complete a focused questionnaire with the patient that was asking specifically about sushi and then even more specifically about tuna sushi. So I called the patient back. The initial interview, I had not gathered a lot of food history because it was a urine specimen, and it's hard to know the right time frame to gather a food history for those. And as I was talking to the patient, I was getting more excited as we went down the questionnaire, but I got really discouraged when the general sushi question, the answer was no. But being a good epidemiologist, we followed through and answered every sub-question, even if the original answer was no. And when we got to the spicy tuna roll question, the patient paused for a minute and said, "Oh, you know what? I actually did. An acquaintance had bought spicy tuna rolls and I tried one." And it was at that moment, this outbreak we were well into it, and the hypothesis had already been established that it was associated with raw scraped ground tuna that was often used in spicy tuna rolls. So when the patient did confirm that they had consumed spicy tuna rolls, a light bulb just went on with me that how cool is this that we can know by the DNA fingerprint of the bacteria in the urine specimen from this patient that it was highly likely that they had eaten this raw ground tuna. And that was just such an exciting aha moment for me about how this information could be used. So within a few months after that, I had the opportunity to move into a specific foodborne role, and I've been working with PFGE and sequencing data ever since. So many of you, if you've been around for a while, know that sequencing or technologies have changed. So the standard for over 20 years for subtyping enteric organisms was PFGE, or pulse field gel electrophoresis. And that would generate these DNA fingerprints of the bacteria that could then be uploaded to CDC's PulseNet system and get assigned pattern names. And then matches of these patterns would trigger cluster investigations. Whole genome sequencing is what has come along and replaced PFGE as the standard. And it analyzes the organism's entire genome, whereas PFGE was only looking at a small sections. And it is more definitive than PFGE and gives a better ability to resolve differences between organisms. And it's also a cost-effective way to get additional information about things such as serotype, virulence genes like the shigatoxin genes, as well as antibiotic resistance. So the granularity of information that you can gain goes on a pyramid like this. So if we know at the base organism level that we're dealing with salmonella, that gives us one piece of information. If we know the serotype, that it's a Salmonella enteritidis, that gives us a little bit more. The PFGE pattern gives more discrimination, and then the sequencing information at the top of the pyramid gives us the most detailed and granular level of information. So as an example, if we had a suspected Salmonella cluster, everything in our dots, each dot represents a case, and we know that they're all Salmonella, so everything looks black. We can't tell any differences between these cases to know whether a subset of them are related or whether they're all related to each other. When we did PFGE, we might see that the PFGE indicates that there's two separate clusters going on, the blue and the teal color. If we did whole genome sequencing on these organisms, we might see that it actually can be broken down further into 3 clusters. But what we found is really important to integrate is that your epidemiologic, traceback, and other evidence can come in and say, well, even though whole genome sequencing might break it down a little bit further, that it's still really important to look at your epi evidence and other lines of evidence to see whether cases are related. So I talked about the tuna outbreak was my first aha moment of how we could use PFGE and subtyping to define outbreaks. My second aha moment came with regards to whole genome sequencing. So in April of 2018, we had our lab here in Washington reported a cluster of 2 Salmonella 14512I- isolates that matched by PFGE. At this point, we were still using PFGE as our main cluster detection method, but whole genome sequencing was being run in parallel. The data was just not being actively analyzed and used in the cluster detection yet. So this particular serotype of Salmonella is one of the top 5 most common in Washington, but despite being a common serotype, this particular PFGE pattern was rare, and it had actually only been seen twice in the previous year of 2017 in Washington. So we had these 2 cases reported in April of 2018. Most of the cluster detection is time-bound, usually somewhere around 60 days is what the lab would look for. So when we looked back in time, we had these 2 closely related isolates in April of 2018, and then back in 2017, in August and October, were 2 additional isolates. So the question became, should we look at this as a cluster of 2 cases in April of 2018 or a cluster of 4 cases stretching all the way back to the previous fall? And so at this point, we decided to look at the sequencing information to see if that could give us any more detailed information that might help answer that question. And so our 4 isolates there are in red highlighted. You can see the 2017 isolates as well as the 2018 isolates. And they all clustered tightly together on the whole genome sequencing tree. And that gave an indication that maybe we should be considering this as one ongoing cluster versus one or 2 separate events. So also when we looked at this tree, I noticed a isolate there right in the middle of our 4 and wasn't quite sure what the story was with that one. Why do we have this extra isolate in the middle of our cluster? And so I was able to look up what that isolate was and found that it was actually from the same county as our 4 current cases, and that it had actually been marked as a different PFGE pattern. So this is one of those instances where the PFGE patterns and the sequencing data don't always have 100% agreement. You can have different PFGE patterns that might be off by 1 band, so they get a different name. But when you look at the sequencing, they look like they're highly related. So we hadn't flagged this one because it was a different PFGE pattern. And looking more closely into the isolate, I found that it had been linked to a previous rotisserie chicken outbreak. So seeing that rotisserie chicken outbreak isolate in the middle of our cluster here really jump-started our hypothesis generation. And so we started to ask further of the cases, had they had rotisserie chicken? Had they shopped at the same chain that had been associated with the previous rotisserie chicken outbreak? And indeed, through investigations, we found that those 4 cases from April and then the previous August and October, indeed all 4 of them shopped at the same location of the chain store. During this time, as the investigation was occurring, we had an additional 3 cases reported that matched. So you can see the 2 cases in June and 1 in July. So we had a total of 7 cases spreading out over almost a year, about an 11-month time span here. So the investigation continued. There was extensive environmental investigation on site. The local health department did an amazing and intensive job investigating on site. And there were a total of 7 cases identified in this outbreak, and all 7 people who got sick shopped at that particular location of the store, but they didn't all buy or eat rotisserie chicken. What the local health department found was that 5 of the cases bought ready-to-eat food from the service deli. 1 was an employee there, and the food history could not be verified for the 7th case. So there was no common food item, but multiple food items from the deli consumed. So then the next question is, we know that this outbreak had occurred over almost a year time span, and all these interventions were put in place based on the environmental investigations. So how do we know that it's actually over this time if it occurred for so long? So this picture here is a current SNP that I grabbed just last week from the NCBI pathogen detection portal, looking at our cluster, and you can see there's still 8 isolates on our little outbreak clade there. 7 of them are the isolates from this particular outbreak, and then that 1 isolate from the previous rotisserie chicken outbreak that jump-started us with our hypothesis generation. So based on this, we can have a good idea that we're not continuing to see transmission in this facility. So another example of how we can use this data is to track over time specific questions. So one question we might ask is, have we seen any recent listeriosis cases associated with an ice cream production facility. So in Washington, we had 3 known human illnesses in 2014 and 2015 associated with ice cream from a production facility. And the 3rd case actually occurred a year later than the original 2. And we know that listeria can persist in the environment for many years. And as we're learning more about listeria outbreaks, outbreaks are being found that span many years time frame. So the question is, we know we had this outbreak in 2014, 2015. How can we keep an eye on and make sure that this persistent strain that had been in the facility didn't come back at some point? So this is, again, a recent SNP of the NCBI pathogen detection portal showing all of the unhighlighted isolates. All the blue ones are environmental and food isolates from the ice cream facility. The 3 red isolates I just found by searching for clinical as the isolate type, and immediately 3 cases pop out. Those are the 3 cases that we knew about, and we can see that there's only 3 clinical on this giant tree, and that gives us good confidence that we haven't seen any other clinical matches to this strain that was in the facility. Now, it doesn't rule out that there could be a new or different strain in a facility, but it does give you a way to track over time known strains. So that's some of why we care about this data and why it's so important to the work that we do. So now we'll go take a step back more into the weeds on how it actually happens. So this is our foodborne team in 2019 at the Halloween costume contest. This was our entry. We did win as the phylogenetic tree and the bases. Unfortunately, we don't have a 2020 version, so we'll have lots of time to plan for our 2021 costumes. So when we're talking about sequencing, we're talking about DNA, which is composed of 2 subunits, the sugar phosphate backbone, and then the nucleotides A, G, T, and C. So we're talking about sequencing actually determining the order in which those 4 nucleotides are strung together in the genome of the organism. So lab work is an ever-evolving field. The first sequencing to be in use was called Sanger sequencing, and it was developed in the 70s. And it was really effective and accurate for sequencing short sections, maybe 300 to 1,000 nucleotides, but not cost effective for large-scale sequencing. Originally, it was done using radio-labeled nucleotides, and then that was replaced by using fluorophore-based methods, so that methodology did evolve over time. This is a chart from NIH showing the cost per megabase of DNA sequencing. If you're not up on your prefixes, megabase would be 1 million bases. So the cost for 1 million bases of DNA sequence back in 2001 is over $5,000. And you can see that it has dropped precipitously. This is a log scale, so we're down below a penny in 2020. So pretty dramatic. And you'll see that the trajectory changed around 2007 to 2008. And the white line there is just an expected rate as technology evolves. Costs go down, but you can see that there is a rapid departure from that expected rate, and that is where sequencing moved from the Sanger sequencing to next generation sequencing methods. So what is next generation sequencing, or NGS? It's sometimes also called high throughput sequencing, and NGS is the technology that is used for whole genome sequencing. There are multiple different technologies. Short read technologies include Illumina and Ion Torrent. Illumina is the most common technology in public health laboratories today. There's also long read sequencing PacBio and Oxford Nanopore. So it can be really easy to think that we send the lab the sample and out pops a lovely phylogenetic tree for us. That's not really what actually comes out of the machine. So after the scientists put the sample on and run, what actually comes out of the machine is more like this, a big pile up of a whole bunch of bits of DNA sequence. So there are a lot of steps that have to happen in the process to go from your bacterial culture plate through extracting the DNA out of those bacteria, shearing the DNA up into little bits so it can be amplified, and then running through the sequencing machine, and then spitting out all those little reads of the DNA sequence, which then has to be put back together so it can be turned into phylogenetic trees, distance matrices, and other ways that you might consume the data. So the challenge is to take those bits, short sequences of DNA that I showed you are called reads. So the challenge is to take, the instrument will spit out thousands and thousands of these short reads, and those then need to be stitched together in the right order to build your genome sequence. So there's two approaches to doing this. One is called reference guided, where you match all of your short little sequence reads up against an identified reference genome, and de novo, where you stitch them together. So you're doing the puzzle without matching against a reference. So this is an example of reference guided assembly. So in red along the top, you see your complete reference genome, and this would be a closely related genome. So you wouldn't use a E. coli if you were sequencing a salmonella. You would pick a closely related organism that you use as your template to match up all of your little reads against the sequencer to where they match up along the genome. And then from all those little reads, then you can stitch together the sequence of your organism along the bottom there. And you can see that there are places where your sequence along the bottom, it differs from your reference. This is expected. You wouldn't expect your salmonella that you're looking at would be identical to your reference one. One concept you might hear is about coverage or depth of coverage. So at this particular point there where you see it 3 times coverage, you can see that there's 3 reads that have a G at that particular position. And so this is one of the many quality metrics that scientists use to assess how good the sequence is. So you would have more confidence in a particular base that has more coverage. So 3 times coverage would be pretty low. You would want to see 20 to 30x coverage or more. So there's a lot of quality metrics that your scientists are looking at to assess whether the genome is good enough quality to analyze. So if you ever get a report back from the lab that they need to rerun a specimen, this is just one of the many quality checks that they're doing to make sure that they have good data to provide you. So how does this actually happen? As much fun as I had using paper and scissors to do my own little assembly here on my whiteboard, this is not how bioinformaticians are assembling your genomes. I would recommend the reference there on the slide actually walks you through how to manually do your own genome assembly like this as a learning tool, and it's actually a pretty fun and helpful exercise to go through. So if they're not using paper and scissors, what are they using? They're using a lot of tools, individual tools, software tools, and then stringing them together into pipelines, which are just procedures that are linked one after the other to accomplish your task. So if you can imagine, if you're trying to sequence a lot of organisms and you're doing each one of these 7 steps one at a time and then manually moving the data through, it would take a lot of time and a lot of work. But what they do with pipelines is a pipeline, once they have built and validated it, it will take the output of the first step and then automatically push that in as the input of the next step and move the data through all of the necessary steps. So far more efficient than paper and scissors, if not quite as much fun. So once you actually get your genome put together, there are a couple different ways that you can look at the data. One that you may have heard of is single nucleotide polymorphism, or SNP. This is good for high amount of resolution for strain comparison, and you can identify where exactly along the genome those SNPs are identified. The other one is multi-locus sequence typing, or MLST, and this allows for developing strain naming conventions that are easier to communicate. One example of this would be the allele codes from PulseNet. And then MLST can either be done on the whole genome or a selected set of genes. So we're going to delve a little more deeply into those analysis methods here. So first, talking about SNP analysis, we have 4 sequences here, A, B, C, and D, and we have them all lined up together. And it's important that you line them up and get them aligned so that you're matching the right sections against each other. And then what we're going to do is we're going to compare each sequence to every other sequence. So we're going to start by looking at sequences A and B. And if you scan through those sequences, you'll find that there are 3 positions or bases where those sequences are different. So if you've seen distance matrices from your lab, this is how those can actually be calculated. So you can see the zeros across the diagonal, because if you compare A to A, it's going to have no differences. B to B, C to C, D to D the same. So in this instance, we're comparing A to B and B to A, and we found that there were 3 differences. So we would fill in a 3 on the table there. If we compare A to C, we also have 3 differences. If we compare A to D, 3 differences as well. Now we'll go compare B to C, only 2 differences. B to D, 2 differences as well. And then compare C to D, and they are identical. So by doing this, we have completely filled out our distance matrix, which then you can look at to get an idea of which sequences are more closely related to the others. If we're looking at MLST, the biggest difference is that your unit of analysis is a gene instead of a single nucleotide. And different sequences for a single gene are called your alleles. So in this instance, we have 3 different alleles at this one gene. And it's important to notice that the allele naming just goes on when the alleles are identified. So allele 1 and 2 are not necessarily more related than allele 1 and allele 45. Each one just gets its own name as it's identified. And with MLST, we can build a distance matrix just like we did with the SNP differences, but by using allele differences instead. So when we're looking at MLST analysis, it lets you look at a subset of the genes instead of looking one by one at every nucleotide like SNP analysis. And just for some scale of numbers, Listeria monocytogenes, looking one by one at nucleotides, you have around 2.9 million. So that's how many spots you have to analyze. whole genome MLST, there's about 4,804 genes that would be looked at. And core genome MLST, you would be using 1,748 of the genes. So I want to go through a visual representation of what the difference is between looking at things a single nucleotide at a time versus by genes. So first we're going to do SNP analysis on our two patterns here. So these are representing our DNA sequences just with stars and pentagons instead. So if we look at sequence or pattern A and B, we'll see that there are a total of 3 characters between them that are different. So we would call this 3 SNP differences. If we then divided, so these are the exact same, not changing the patterns at all, but just breaking them into their own little gene houses here. So with MLST, we're looking at a gene level instead of individual level. And all we care about is whether they have the same sequence within each little house here. So if we look at allele differences, you can see that in the first house there, that there's a difference. The second 2 are identical. The third, we have different patterns. The fourth is identical. So this would be a total of 2 allele differences between these 2 sequences. And then if we add on the coloring for our SNP analyses, you can see that there's 1 SNP difference within that first gene, but there's 2 in the second gene. So we have a total of 3 SNP differences, but only 2 allele differences. Because when you're looking at the alleles, you're just looking at a yes, no, same or different versus how different. So it's important when you're looking at distance matrices to know the analysis method that's being used. So these are both from our pattern on the previous slide, and you can see that you get a different answer whether you're using SNP or MLST. So it's important to understand what you're looking at. So now we have our information, and we want to take that distance information or that sequence information and turn that into something a little more pretty. And so we're going to talk about going from nucleotides to trees. So what do phylogenetic trees actually do or show? So phylogenetic trees can help infer the relatedness between isolates and then approximate the evolution of the pathogen over time as it's reproducing. So we're going to look at 2 sequences here, sequence 1 and 2, and you can see the 2 different colored bases. We have, there's 2 differences between these sequences. So the question is there a sequence that would be a potential common ancestor to both of these sequences that would not have to take a lot of steps to get to either of these sequences? So we can see that everybody starts with CTG, but in that 4th position, our ancestor sequence there goes from a C to a G to get from the ancestor to sequence 1. And then to go from the ancestor sequence to sequence 2, it also changes by 1 nucleotide, this time going from an A to a C. So the theoretical sequence that we put up above our 2 sequences is a sequence that it only takes 1 mutation or nucleotide change to get either to sequence 1 or to sequence 2. And if we move this around to put it in what you might used to be seeing as phylogenetic trees, you can see we have our inferred most recent common ancestor sequence that takes 1 mutation difference to get to sequence 1, or a different mutation, single mutation difference will get you sequence 2. So let's talk about some tree terminology that you might hear. Out on the edge there, so we have A, B, C, and D, and those can be called a few different things. You might hear leaf, tip, or node. And then at the intersection between A and C, those are called internal nodes. And then the branches are the horizontal lines connecting your isolates. And what's highlighted here is a clade or a monophyletic group. And it's a group of your leaves, tips, or nodes that are descended from a single common ancestor. And one way that you can identify a clade is that it's a piece of the tree that can be chopped off by just making one cut. So if you just chopped the tree at this point here, this whole clade would fall off. And you might be noticing that there is another clade within that clade. So clades can be nested within each other. So we're going to try and take it from theoretical to a little more example-based of how we think through outbreaks. So this is a demo of salmonella-contaminated pork, something we've dealt with in Washington. So we have down at the bottom, we have a pork farm that has some salmonella contamination, and that pork has been shipped to multiple locations and ends up on these two dinner plates. On the left, we have 3 people consuming the pork together. On the right, we have 3 people also consuming the pork together. And our little bacteria there indicate that 5 out of the 6 people become ill with the salmonella from this contaminated pork. And if we kind of just reframe those lines that we saw tracing the movement of the pork, the contaminated item, through the food chain from farm to meal to person, and just square those off, you can see that it starts to look more like a phylogenetic tree. And if we turn that around, so all we're doing is rotating our tree here, then you can see even more clearly that we're really building a phylogenetic tree of the salmonella as we go along. So how do we relate trees and distance matrices? So we learned about branches, and the genetic change that you see here marked is the distance. When you're looking at a tree, only the horizontal distance matters. The vertical distance is just for spacing out the tree for visual purposes. So if you want to count or look at how much genetic distance is between 2 of your isolates, you'll need to sum the distance of the horizontal branches on your tree. So in this case, if we wanted to look at, we'll say that this is a SNP-based tree. So we want to look at the distance between isolate C and B. So you would sum the first branch there is a distance of 6. So you would sum 6 plus 1 plus 3. So our distance between C and B is 10. And if you wanted to look at the distance matrix over here, we would look for the distance between C and B, and we can verify that it is indeed 10. So depending on how the tree is built, it can be represented as SNPs, it can be represented as substitutions per site or some other ways depending on the analysis. So it is important to look at your scale and understand how that's being measured. But in general, it's the horizontal distance that matters. So the longer it takes you to travel horizontally between 2 isolates, the further they are from each other. And you may see, so we have our tree here, we have our distance matrix, which we talked about how those get calculated. You may also see down below the distance matrix what's called a heat map. And that's just a color-based way of representing your distance matrix. So you'll see across the diagonal there is all of our zeros from our distance matrix above. So those are the blue. And then the dark red indicates the furthest distance from each other. So you can see that down in the bottom right, we have dark red, and those correspond to the 19s in our distance matrix. So this is just a visual representation of the distance matrix. So one thing to note about trees, we mentioned that only the horizontal distance matters, not the vertical distance. So you can actually flip the tree at any of your internal nodes, and it means the exact same thing. So if we go back, we can see here we have C on top, and then A, and then B below it. But if we flip that whole clade around, where we have C on bottom, and then A, and then B, if we do our summing of our horizontal distances, you'll see that it's still 6 plus 1 plus 3. It's just a different way of building the tree. So trees can be, most often in the foodborne world, we tend to see trees lined up the way that I showed above, but trees can be looked at different ways. You can have a radial tree, you can have it horizontal, or you can actually flip the whole tree and have it vertical. One thing to note as a caveat to my previous statement, if the tree is flipped horizontally, as in this one in the bottom right, then your branches are now the opposite way, so your distances would be measured vertically, not horizontally. So there are limitations to trees. The sequence specimens or isolates only represent a fraction of the actual cases, not a true reconstruction of everything that happened along the way. Strains that are phylogenetically closer are more likely to share an epidemiologic association, but that's not set in stone for every case. Outbreaks may or may not be clonal. We've seen, for example, the Salmonella outbreak in Kratom was caused by many different serotypes and strains within those serotypes. And then the integration of the epidemiologic evidence and environmental health and traceback data is really needed to best interpret trees. So one of the critical questions is, how close is close enough? So allele or SNP differences within a cluster may be larger or smaller depending on the particular organism, as well as other characteristics of the cluster or outbreak. More clonal species or serotypes may have a smaller allele difference. So we see this a lot with Salmonella enteritidis, where it's very clonal and tends to have a very tight allele difference or SNP difference between isolates. Zoonotic outbreaks may have larger allele or SNP differences. We've seen this often in the live poultry outbreaks. And the epidata and environmental health and traceback data is critical to help define clusters and thresholds. So we're going to look at a few outbreaks to talk about how you can look at trees and try and make sense of what they're saying here. So all of our isolates in red here are from a confirmed Salmonella Newport outbreak. And so we can see a very clear defined outbreak clade here, where our isolates of interest are not only grouping together, but they're grouping together by themselves to the exclusion of all the other isolates on the tree. So that's a very pretty, nice clade for an outbreak there. It's important when you're looking at a tree or a clade to pay attention to the scale. So you can see this was another outbreak, all the isolates in red. You can see most of them were 2019 isolates and then one early 2020 isolate. And if you look at the tree there, you might think, well, maybe this new unhighlighted isolate there is related. Maybe this outbreak's ongoing. If we look at our tree scale there, this is measured in SNPs, and you can see that that isolate, even though it falls within the clade with your other isolates, is actually 20 to 30 SNPs difference from your other ones. So taking that combined with the temporal difference would make me less concerned that this is a highly related organism. So another example is a listeria outbreak that was found to be in finished ice cream. So this is from a FDA paper, and this is where they were trying to tease out this particular ice cream facility had previously had issues with listeria. So they were trying to tease out whether the outbreak was coming from something within the facility or whether it was coming in on a raw material, and that raw material was being carried through and causing the outbreak rather than something that was happening within the facility. So all of the blue isolates there are from a particular supplier of this ice cream facility. The green ones are actually from the facility itself. And you can see that the isolates are all interleaved within each other. They all cluster together, but there's no clear differentiation between the ones from the raw material and the ones from the facility. So this supported the investigation conclusion that it looks like this particular listeria was coming in from the supplier and then being found in the producer. So here's another outbreak, and just one thing to note here, you can see the median was 17 SNPs and a range of 0 to 23 SNPs. So not a super tight range there. So here's another example. This was I believe a salmonella, and the red ones were a salmonella cluster that was detected in 2017. And it was found that they were 13 to 14 SNPs different from 3 isolates found in a food production facility in 2012. So the question was, should we consider, is it likely that these 2012 food production isolates are related to this 2017 cluster that's being investigated? And you can see based on the just looking at the SNP numbers that might be something to look into. But also by looking at the shape of the tree here, you can see that though they group together in one large clade, they split out pretty cleanly into 2 separate clades where your facility isolates group together and then your 2017 clinical isolates group together separately. And based on the shape of the tree, the time difference, and not finding any epidemiologic link between this cluster and the food produced at that facility, it was determined that these were not likely to be causally linked. So some final takeaways. The laboratory and bioinformatics work that it takes to go from the test tube to the phylogenetic tree is significant. Appreciate your laboratorians, your bioinformaticians, and all the work that they are doing. I know it can be hard to be patient. I think epidemiologists particularly can get excited and want results now, but I hope you have some appreciation of all the steps that it takes to go through to get that final output. You don't need to be able to run bioinformatics pipelines to be able to interpret trees. Work closely with your laboratory and bioinformatics colleagues and ask a lot of questions. cluster and outbreak detection requires lab, epi, and EH evidence to come together to tell the whole story. As powerful as whole genome sequencing is on its own, it can never solve an outbreak without the other disciplines working in concert. And finally, don't be intimidated. Keep finding opportunities to learn, whether they're at conferences or webinars. Don't be afraid to take courses that you maybe feel like are above you. I find that with this topic, you have to keep taking. It's like getting a kid to like Brussels sprouts. You have to keep trying it over and over before it clicks. So I hope that after going through all this, I hope that you're more comfortable with some basic understandings, and that if you get something from your lab, like there's a cluster of 5 cases within 0 to 10 alleles, that makes sense to you, that you would pick out that since they're saying 0 to 10 alleles, that they're using MLST analysis there, and that you can understand the data that's being communicated by your laboratory. So here's some additional tools and resources. The New York Food Safety Center of Excellence has a couple live learning series recordings, as well as a whole genome MLST activity. And then APHL has a couple webinars. The cluster detection one is more focused on the lab and using their bionumerics programs, but the cluster detection and reporting 2 is a little more general about how to use epi and lab data to detect and communicate about clusters. And with that, we will move on to Q&A time with Beth.
Great. Thanks so much, Cassandra, for a really nice presentation. I am going to start reading questions from the Q&A. So the first one, Cassandra, are there advantages or disadvantages in using SNP versus MLST to describe differences in genomes?
There are. I think CDC and PulseNet have leaned towards using MLST because it's more easily collapsed into things like allele codes that can more easily be shared.
Okay. Second question, can you define clonal? I always have a hard time explaining to non-laboratorians what this means in simple terms.
I think my best definition would be descended from a common source.
Okay. And then currently with WGS, we depend on culture isolates. Do you think someday we will have tests like WGS to use on non-cultured samples, for example, if it's PCR or other CIDTs?
Yeah, that's a great question. So you hit a critical nail on the head here, that to do this work that we're doing that's so critical, it does currently require isolates, and with all the culture independent diagnostic testing, we're missing out on getting those cultured isolates. So there is a lot of work being done looking at other methods like metagenomics, where you can do it not having the isolate. So it's not in wide use, and we're not there in public health, but there are options in the future.
All right, thank you for your comment, Richard. Here's another question. Are there certain genes or nucleotide sequences that are unlikely to change between bacteria that would indicate closer or more distant relation if they matched or did not match? Essentially, is there a qualitative difference between genes that indicate similar serotypes, or is it purely quantitative in determining relation?
I think my best on that one would be the the core genome versus the whole genome MLST. So the idea between using the core genome is that those are the common genes to most organisms in that species. So that's the idea between using the core genome versus the entire genome.
Okay, do you have thoughts about absolute versus multiplicative scales on phylogenetic trees? I have sometimes seen percent difference as the scale instead of number of SNPs.
Yeah, I don't have strong opinions on that. I think it depends on the ways that the trees are built, and I think it's a little beyond my technical skill to address.
Okay, here's another one. Are there ways to consider changes over time when evaluating phylogenetic trees? For example, supporting use of a flexible temporal window in defining a cluster?
I think we definitely need to be looking at how we're defining clusters, both looking at them on trees and in just distance matrices and other ways, because we have seen that clusters can carry on longer than our kind of standard 60 or 120 day windows.
Okay, I think that's all the questions I have so far. We'll just give it a minute because we have a couple minutes left. Here's one. How is the degree of relatedness determined from one organism to another? So does one organism have greater variability in what we may call a cluster than another?
Yeah, so each organism is unique, and then each serotype or other sub-designation is unique as well. So what works as a cluster definition for Salmonella Newport might not apply as well for Salmonella enteritidis. So it's still very much a learning process of what constitutes a good cluster definition.